Hello and welcome to GCSE Biology 9 to 1. Today's topics are light microscopes and staining. Light microscope. So here we have a picture of a light microscope. So what you will be able to learn today is the parts of the light microscope and why they're used. So a light microscope is used to examine specimens that are still too small to be seen by the naked eye. So we have to use a light microscope to see them. So today we'll start with the parts of the light microscope. As you can see here, this illustration has the parts of the light microscope. So you will maybe be asked to remember the parts of the light microscope, and you might be asked to label them. So here we have some of the important parts of the light microscope. As you can see, we have the eyepiece lens. This is where you put your eyes to look at the specimen. Then we have the coarse focusing wheel and the fine focusing wheels. You would use these to control the magnification and to get a better view of the image. Then we have the objective lens, which is the piece that lets you see the actual specimen that goes through the eyepiece lens. Then you have the stage. This is where you put the specimen, and we have the light source, which illuminates the specimen. The light source is needed to see the specimen better. So those are the basic parts of the light microscope. As you can see, we have the cover slip here, which is where you place your specimen, and you also have another part of the cover slip where you put on top of the specimen. So the cover slip is used to protect the microscope and prevent the slide from drying out when it's being examined. So for example, if you get an onion cell, which you might use in a practical, you have to place it on the cover slip and put this on top of it to prevent it damaging the objective lens, and also make sure it's not dried out so it doesn't damage the actual specimen. As you can see, this is just a fact. Hans Janssen created the first compound microscope in 1590. So it's been here for quite a while, and we now have more advanced microscopes such as electron microscopes and transmission electron microscopes, which I might cover in a later video, but today we'll be focusing on the light microscope. So staining. Staining is a technique which cells or thin sections of biological tissue that are normally transparent are immersed in one or more colored dyes to make them more visible. So stains are used to get a specimen, for example, like I used before, the onion cell. Without a stain, you won't be able to really make out much of the cell. You won't be able to see the details of the cell. So what you use a stain, for example, methyl blue. You might use this one when you're doing the practical to stain the onion cell or the specimen you're looking at. Then you'll be you will look at more clearly to see the different parts of the cell and the surrounding cells. For example, here in this picture, we have an onion cell. As you can see, this is the nucleus and these are the cell walls. Well, this is a plant cell. I will cover plant and animal cells also in a later video, so make sure you check that out. So also another important thing to remember is staining heightens the contrast between the various cells or tissue components. This means that you will be able to make out the different parts of the cell more clearly. So here we have some maths. So this maths of part of this section isn't too difficult. So this part is talking about the calculation of total magnification. So this is a relatively easy calculation. So as you can see, this is the equation. The total magnification equals the eyepiece magnification times the objective lens magnification. So if you remember this, you should be able to answer the questions using this calculation. Now I'm going to use an example here. So for example, if the eyepiece lens is 10 times magnification and the objective lens is 4 times magnification, the total magnification will be 10 times 4, which will give you 40. As you can see, the objective lens normally has it labeled here what the magnification is. So this is a different calculation. This is called the calculation of magnification. Now, if the question is asking about the calculation of total magnification, you will use the equation given before. But if the question is asking about the calculation of magnification, then you will use this equation. As you can see, this diagram is called a formula triangle. And as you can see, this has the different parts of the equation. Now, this is an easier way to remember it rather than remembering the full equation, as you can use this to work out what parts you need for the equation. So for example, if they ask for the magnification, which is most commonly asked, they normally ask for the magnification. What you do is you cover the magnification and then you look what's left of the equation. So to work out magnification, you will need the size of the image divided by the actual size of the object. That is how you work out magnification. Now I'm going to show you the methods of working out actual magnification. So for this one, you have 3 steps here. It's relatively easy. So what you do here, we have an example of an exam question. Here what you do is you measure the scale bar. So as mentioned, it's beside the drawing and it's in millimeters. So measure it in millimeters. So what you do is you use your ruler to measure the length of this part right here. This is what we call a scale bar. So you measure that in millimeters. And then you convert it to micrometers by multiplying it by 1,000. So you multiply that value by 1,000. Then, to find out the magnification, you use the scale bar image and divide it by the actual scale bar length. So the actual scale bar length is this part right here, which says 2 micrometers. So you divide the length of this measured in millimeters, then times it by 1,000, and then divide this by the actual scale bar length. The best way to do this is use this triangle and practice for yourself by using some of these questions right here. And I'll put down the actual answers to this so you can check later if you understood the calculation. So in summary, you should be able to label parts of the microscope, explain the use of cover slip, explain the use of staining, and calculate total magnification, and also calculate magnification. So if you have any questions, let me know in the comments. I will be uploading more GCSE biology videos later on. So thank you for watching and make sure you like and subscribe.